Hello, my name is Lucas Rodrigues de Mello, and today I will be presenting the work A Biophysical Approach for the Characterization, Development and Delivery of Nucleic Acids of a New Cell-Penetrating Peptide for the annual Brazilian Biophysical Society Congress poster session. Firstly, what is a Trojan peptide? A Trojan peptide, or a cell-penetrating peptide, usually is a short and cationic peptide with the capacity of translocating through cell membranes. As the Trojan horse in the Greek myth, those peptides can be used as carriers for other molecules inside living cells. Those molecules can be proteins, biomaterials, CPPs can be used for enhancing the uptake of poorly absorbed drugs, or for the internalization and delivery of nucleic acids, which is the focus of this presentation. Due to the cationic nature of these peptides, they are attracted for the anionic DNA and aggregate around it. So it's really easy and cheap to make nanoparticles using CPPs and nucleic acids. And we can use this complex for the delivery of foreign DNA inside living cells, which is a current challenge in the fields of molecular biology and gene therapy. For this work, we presented a minimalistic approach where we take a classical CPP sequence, penetrating, and isolated a small ball amphiphilic fragment, which we denominated Q7. The reason for this is the fact that the synthesis of a small peptide is easier, cheaper, and usually we have a better control of the self-assembly of those small sequences. The aim of this study is to fill a lack of evidence in how penetratin and Q7 aggregates with nucleic acids without any kind of covalent binding. For this, we used a model double-stranded DNA of 200 base pairs co-solubilized with the peptides and conduct a series of structural and biological assays. Here, I will show you some of the data regarding this work. We determined the critical aggregation of Q7 by fluorimetric assays around 0.03 mg per microliter, which is already a difference concerning its mother sequence penetratin that do not aggregate. Determining the macroscopic structure of complexes between penetratin, Q7, and our model DNA, we performed Sachs assays. These assays determined that penetratin highly condenses DNA, denoted by this Bragg peak, while Q7 condensates into a smaller network of fibers with a lesser degree of order compared to penetratin. This hypothesis was confirmed with the use of atomic force microscopy, transmission electron microscopy, where we can observe highly condensated structures between DNA and penetratin, even resembling the condensation of DNA by stone proteins. We also observed by nano-infrared spectroscopy that these complexes are composed by a peptide core with an outer shell of DNA enveloping this peptide. For Q7 conjugated with DNA, we observed an entirely different aggregation pattern, with the presence of smaller fractals in lower concentrations and an intricated network of fibers in higher concentrations. The presence of these structures in solution was confirmed by cryo-electron microscopy. And finally, we performed the delivery of this peptide conjugated with our 200 base pair DNA labeled with YOYO1, a green fluorescent probe, into the cytoplasm of HeLa cell. The conclusions are that both Q7 and penetratin aggregated into nanostructures with distinct properties in the presence of nucleic acids, with penetratin presenting a higher degree of condensation compared to Q7, and that Q7 can be used as a carrier for the cytoplasmatic delivery of DNA. Both penetratin and Q7 resulted in publications, respectively a cover article in the JPCB and a back cover in Soft Matter. I would like to thank my PI, Emerson Rodrigo da Silva, and all the team of LabEst from Unifesp. We'd also like to thank the University Unifesp and the funding agencies FAPESP and CNPq for funding this work.